Assalamu alaikum. Welcome you all in this online class. I am Imran Ahmed and today we have a class on cell division from chapter 2 of class 8 science book, and today our topic of discussion is cell division, its type and process of cell division. So from this topic all you can gauge the idea what we can discuss today, what we will going to discuss today. Cell division. So at first, what is cell? All you know, cell is the structural and functional unit of living organism. All you know that. And division, what is division? Division means to divide something into two or more than two parts. This is cell division. And all you know, the life of a living organism initiates from a single cell, and at the passage of time this single cell develops multiples of cells, billion trillions of cells, and say an organism of trillion of cells looks very gigantic, but the initial stage it's were single cell. So how this single cell convert into trillions of cells? This is through the cell division. So if you want to define what is cell division, so we can say the process by which one cell divides to form two or more than two cells is called cell division. Okay, and the types of cell division here we can see different types of cell division in different organism. In lower organism we see this amitosis type of cell division, and in higher organism we can see mitosis and meiosis, these two type of cell division into different phases. Okay, so amitosis cell division. This amitosis cell division is also known as direct cell division. Why it is called direct cell division? Here we have a figure of amitosis cell division. Here we can see this is a cell. Suppose this is a primitive cell like bacteria cell, and here inside this cell we have nucleus. This nucleus contains the genetic material, and before this the cell and of its nucleus elongates and become dumbbell and shaped, and the genetic materials without any complex process divides into two equal halves, and the outer covering forms a furrow, this furrow, and in later stage this furrow gradually increases towards each other and the nucleus divides into two. Nucleus, that means genetic material, divides into two, and as the whole genetic material divides into two, then the outer membrane touches each other and it forms two daughter cell. In initiation or primary phase the daughter cell becomes very small inside, but in the passage of time it becomes large inside, that means it gets its normal shape, and here we say this is daughter cell. So why we call these two cell as daughter cell? All you know, the cell which is produced by this division, and after some time this cell again participates in this division and again it gives two daughter cell and two new cells. So in this universe only the daughters, females, can able to give new cells or new offspring. That's why we call this daughter cell, and as the cell divides without except any complex process, that's why this is called direct cell division. So amitosis cell division is called direct cell division. And where this amitosis cell division occurs? At first I say this amitosis cell division occurs in primitive type cell. All you know what is primitive type cell and which organisms bears this primitive type cell, and these are, as example, we know the bacteria in several yeast, amoeba amoeba. This amoeba belongs to kingdom Protista, but here in this amoeba we see this amitosis cell division. That's why we mention this name here. So these are the example, and this is the process of amitosis cell division. As it divides without any complex process, that's why it is called direct cell division. I hope all you will get what is amitosis cell division. And the second type of our cell division is mitosis, and the third one is meiosis. So today we mainly discuss the process of amitosis and mitosis cell division. So now we have the mitosis cell division. So what is mitosis cell division, and where does this mitosis cell division occur? So at first, if we want to define, mitosis cell division occurs in the somatic cell of living organism. Somatic cell of living organism, and this type of cell division occurs in the growing portion of plant, like meristem tissue, in the apical portion of root, in leaves, and in animals in somatic cell. This mitosis cell division takes place. Okay. So the process of cell division in which one mother cell, suppose this is a mother cell, this is a mother cell bearing 2n number chromosome, and it gives 2 daughter cell with same characteristics and same number of chromosome, 2 daughter cell. So this is the mitosis cell division, the process of cell division in which one mother cell gives or produces 2 daughter cell with same characteristics and same number of chromosome as its mother cell, is called mitosis cell division. If we want to define this mitosis cell division shortly, then we can say the process of cell division in which the nucleus and chromosome both are divided only once and produces 2 daughter cell is called mitosis cell division. As here the chromosome divide only once, that's why the characteristics remain same as mother cell, in daughter cell as mother cell. This is mitosis cell division. And mitosis cell division divides into process, happens into 2 phases. One is karyokinesis, and another one is cytokinesis. In my diagram, I try to show all the things what is happens in this mitosis cell division. Suppose in a cell goes to divide through mitosis process. So suppose this is the cell, this cell goes to division through mitosis process. So this is normal cell, and when this cell goes to divisionary phase, then it needs some preparation. Here the chromosome divides once, so this division takes place in this phase, in this first phase or initial phase. This phase is known as interphase. This is a phase in which the cell prepares itself for the next division. This is called interphase or preparatory phase. So what is interphase? If you have question, so then you answer this question like this: the intermediate phase between 2 consecutive cell division is called interphase, or the preparatory phase of cell division of cell is called the interphase, and after that the cell goes the division phase. So the division phase in mitosis cell division happens in 2 phases. One is karyokinesis and another one is cytokinesis. So karyokinesis, according to my previous lecture, all you know what is karyon, and karyon means nucleus. So karyokinesis means the division of nucleus. So division of nucleus takes place in this cell division process in 5 phases: prophase, prometaphase, metaphase, anaphase and telophase. So these 5 phases covers the karyokinesis, and at last the cytoplasm divides. This is known as cytokinesis. So what happens in this cell division? So at first the genetic material duplicates itself in preparatory phase, but it remains in thread like structure. But in prophase stage the nucleus becomes large in size and the water dehydrates from the nucleus, and mainly this water dehydrates from the chromosome, and this water comes in the cytoplasm. Okay. As the water comes out from the nucleus, then the chromosome looks very short and thick, and at this period these chromosomes are easily visible under the light microscope and they can attain the colors. So this is the prophase. This is the incident that happens in prophase stage. The nucleus becomes large in size and the water dehydrates from the nucleus and comes in cytoplasm, and at this stage the chromosomes are visible under the light microscope. Then next phase of this karyokinesis, this is prometaphase. Prometaphase, that means the first phase, initial phase of metaphase. In this phase the spindle apparatus develops in the cell, and the nuclear membrane, here we can see these are the red dotted, this is the nuclear membrane and this is dotted structure is our nucleolus. So in this stage nuclear membrane and nucleolus becomes start to disappear, and spindle apparatus with 2 pole is developed. Here I draw this figure of plant cell. So in plant cell this spindle apparatus develops with the help of microtubules. Microtubules is the cytoskeleton of cell. So microtubules helps to develop this spindle apparatus. This is the pole and this is the pole, and these are the spindle fiber. So the spindle fibers develops from one end to another end and it forms the spindle apparatus. And here we have in prophase stage we see 4 chromosomes. So 4 chromosomes, here the chromosome duplicates but it remain united condition, and here the chromosomes with the help of centromere get attached with the spindle fiber, and the fiber, the spindle fiber which holds the centromere is called the traction fiber. Clear? So the spindle fiber which holds the centromere is called traction fiber. Not all the spindle fiber holds the centromere, only few holds this centromere. Those fibers hold this centromere is called traction fiber, and the rest of the fibers simply known as spindle fiber. Okay. So here the centromere get attached with this spindle fiber, and then this centromere can stay in different position of this spindle apparatus, different position. There is no specific position of this centromere in spindle apparatus. But in next phase, in metaphase phase, all the chromosomes along with their centromere comes at the middle of this spindle apparatus, So this is one type of arrangement. This arrangement is known as metakinesis. What is metakinesis? So if you have question what is metakinesis, then you say the arrangement of chromosome at the middle or equator region of spindle apparatus in metaphase stage of mitosis cell division is known as metakinesis. In this phase the parts of chromosomes, here we see in both sides of centromere. These are the chromatids. Parts of chromosome is known as chromatids. The chromatids becomes more shortest and more thickest. Okay, and at this stage the chromatids which remain in close contact during metaphase and prometaphase, but in this phase they separates from each other. Here we can see these are the shortest and thickest chromatids, and in next phase, in anaphase stage, we can see the centromere splits. That means the centromere divides, and the chromosome previously divides longitudinally, and when the centromere splits, this division of centromere divides the single chromosome into two daughter chromatids or two daughter chromosomes, and this newly formed daughter chromatid moves to opposite pole through the traction fiber. Okay, and in this movement we can see centromere always remains ahead and the chromatid follow the centromere. And here we can see here the arms of chromosome, that means chromatids, both are same in size, but here we can see these are not same in size. So in a cell we see 4 different types of chromosomes on the basis of the position of centromere. Suppose this is a chromosome and this is the centromere, position of centromere, and suppose this is the chromosome and the centromere remains in one side 1/3 and one side 2/3, and in another chromosome the chromosome remains very close to one end, and in another it remains at end portion. Okay, so here during this movement, when the centromere splits and it starts to move the opposite pole, then it takes this type of shape in this movement. As I mentioned, the centromere remains ahead and the chromatid follow these centromeres, and here when it moves during the anaphase stage, it takes this type of shape, this L shaped. This is L shaped and here this is V shaped, and in this case in movement the chromosomes looks J shaped structure, and here the chromatids can't bend, that's why it looks I shaped structure. Here this is J shaped and this is I shaped. So these 4 different shaped chromosomes are named in different way. V shaped chromosomes is known as metacentric, as the centromere present at the middle of chromosomes, and the L shaped chromosome is known as submetacentric, J shaped chromosome is known as acrocentric, and I shaped chromosome is known as telocentric. So during the movement in anaphase stage, the chromosome takes different shapes. These are V, L, J, I: metacentric, submetacentric, acrocentric and telocentric. These all things you will get in your book. Okay, so this is the anaphase. When the chromosomes, daughter chromosomes or chromatids comes near to pole, then this anaphase stage comes to an end. And then when all the chromosomes comes at the polar region, that means in pole, then again hydration takes place. Hydration takes place as water adds here. That's why the short and thickest chromosomes becomes again takes the shape of thread, and again the nuclear membrane and nucleolus reappears here. Clear? So nuclear membrane, nucleolus and hydration, all the things takes place, and in this way 2 new nucleus is develops. So here we can see this is single nucleus, and this single nucleus after 5 different steps it divides into 2 nucleus, and in this division the chromosome also divides and it comes into new nucleus. So in this division, the nucleus and chromosome both are divided once. This type of division is known as mitosis cell division, and as the nucleus divides here, that's why we include these 5 phases: prophase, prometaphase, metaphase, anaphase and telophase as karyokinesis. So karyokinesis takes place through these 5 steps, and last of all, this is cytokinesis. All you know, this is 2 nucleus, but 2 nucleus is present in a single cell. But when the 2 nucleus develops, the spindle fibers, these spindle fibers in case of plant cell touches the cell wall of cell, and the phragmosome like lysosome deposits here, and a cell plate develops here. This is the cell plate. This, after formation this cell plate, the different types of chemical substance, cellulose, hemicellulose, glycoprotein, all are deposits on both sides of this cell plate, and finally 2 daughter cell develops. So when the cell plate completely develops, then it divides the cytoplasm. This division of cytoplasm is known as cytokinesis. So mitosis cell division takes place through these 2 processes, karyokinesis and cytokinesis. Okay. So karyokinesis, division of nucleus; cytokinesis, complete division of cells. That's all. And as here the chromosome divides into once, and nucleus also divide once, so both the nucleus, both the newly formed cell bears the same numbers of chromosome, same characteristics, as the new daughter cell holds same characteristics, same number of chromosome. That's why this mitosis cell division is called equational cell division. Why equational cell division? I hope this is clear to all of you, because in mother cell number of chromosome is 4, in daughter cell number of chromosome is 4. That's why this is called equational cell division. I hope this is clear. And where do this mitosis cell division occurs? We know in case of animal's body it occurs in somatic cell, but some exception is present in our RBC, that means red blood cell. In our nerve cell no cell division occurs. Why? Because no active centriole is present here. Okay. And I forgot to mention one thing: in plant cell, the microtubules, which acts as cytoskeleton of cell, these microtubules helps to develop the spindle apparatus. But in case of animal cell, the centriole which exists inside the centrosome, these 2 centriole take position in 2 opposite poles, and the centriole radiates the aster ray, and the aster ray comes closer and it forms the spindle apparatus in case of animal cell. So this is the steps of mitosis cell division, and the third one is meiosis cell division. In my next lecture I will try to discuss this meiosis cell division and difference of mitosis and meiosis cell division and its importance and the heredity, and the most important thing, DNA. Okay. I hope this lecture will help you a lot, and if the lecture help you, then it seems to me very satisfactory. That's all from my side. All of you try to understand this and try to answer all the questions in your exam appropriately and correctly. Thank you. Allah Hafiz.